let us start discussing now. Now, one very important thing I want to tell to you that you guys are not final year students. Okay. Why am I telling you this thing is that if you guys are final year students, you will be writing the next exam, right? So you guys are, sorry, you will be writing the MCI, FMGE exam. So you guys are not final year students. So you guys are most probably second year, most of them third year, right? So most probably you will be writing the next exam. So when it comes to the next exam, most of the students already know what I'm trying to tell you, that it will be completely clinical point of view. So when I teach microbiology, I don't teach as it is microbiology subject. Rather, I will integrate this subject with some other subjects like pathology, medicine, orthopedics, right? So even you have infections in orthopedics also. So what I will do is that even with physiology also. So I'll integrate this microbiology with remaining subjects. So your friends might have already told because I have already taught microbiology classes for you guys, right? So I hope your friends might have already told. Yes, 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 yes. Very good. So these are the things, guys. So completely the lectures will be clinically oriented, and we are going to, our target is that we'll be completing the microbiology by 15th of January. So what I am telling to you seriously is that if you attend all these lectures, then I'm guaranteeing you that at least 70% of clinical knowledge you will definitely gain. At least 70 to 80% of how you have to answer the clinical questions also you will understand. Okay. So these things are very, very important. So if you are all okay with that, I'll start the classes. You will understand how clinically I will teach you. And most important thing is, you want me to use a PPT and read the PPT, or you want me to write, draw and explain you? Batao jaldi. Yes. Right. So guys, like, tell me that is there any new student over here? Any new one? Yeah. Any new student? So Martina is new, Utkarsh is new, MD phase. Okay. So I just wanted to know how many new students are there. If you are all old students, then that would be fine because you already are in my pace. You understand my style of teaching and all. So that would be okay. Right. Just tell me, am I audible to you? Once again, one last time, a small glitch is there here. Am I audible to you? Yes. Right? Fine. Great. Now let us start discussing these things. So the first important, okay, the first important thing which you would see, not needed, not needed, not needed. So the first important thing which you would see over here, right, is let us start from the complete basics, guys. Okay. So in this picture, you know that this is a picture of the bacteria. I hope all of you are making the notes, right? I want every one of you to make the notes also. Okay. So this structure which you can see, this is the structure of the bacteria, right? Now, within this structure, what are the important things you can see over here? You know that this particular part in the center, whatever I've drawn over here, this is called as your cytoplasm. All of you know this. This is called as your cytoplasm, right? Now, after this cytoplasm, what is that red color lining that is cytoplasm? Tell me, what is that red color line which is lining your cytoplasm? This particular structure over here, this particular structure over here is called as your plasma membrane. Okay, this particular structure over here is called as your plasma membrane. If you want, you call it as plasma membrane, or if you want it, you can call it as cell membrane also. Fine. Right. Now, after that, let me shade one very important thing and you will tell me what is this. Okay. For example, let me shade this particular part over here. Now, tell me what is this particular part which I'm shading with the yellow color, guys? Yes. What is this particular part which I am shading it with the yellow in color? Anyone? What is this? Very good, very good, very good. No, 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 no. This is not called as a capsule. This is called as cell wall. What is this called? This is called as your cell wall. Okay. Now, surrounding your cell wall, this blue color layer you see, this blue color layer is nothing but called as your capsule. Surrounding this cell wall, you see a blue color layer that is called as your capsule. So three things, cytoplasm, cell membrane, surrounding the cell membrane, you have got cell wall, surrounding the cell wall, you have got capsule. Now, one very important thing is that on the surface of the capsule, you have got some structures which are very long, you have got some structures which are very short. Okay. So, ye jo structures hai na, these structures which are very long, like these structures which are very long, these are called as your flagella. This is a complete basic, guys. I know you know these things already. These are called as flagella. And the structures which are short and straight, ये देखो, यहाँ पे structure देखो, this is short and straight, right? So this structure is called as pili or fimbriae. What is this called as? This structure over here is called as your pili or it is called as your fimbriae. ठीक है? So यहाँ तक तो you understood. There is nothing to understand here because this is the same things which you have studied all the time, right? Now comes the important point. What is the important point here? The important point here is that they are going to ask you, pili is made up of what? Pili is made up of an important protein called as glycoprotein. Pili is made up of what? Pili is made up of glycoprotein. Okay, pili is made up of what? It is made up of glycoprotein. So this is one very, very, very important question which you need to understand. Okay. Second important thing. You know cytoplasm, what are the contents of the cytoplasm? We will discuss this later on. You know the plasma membrane. You know the cell wall. Cell wall is the thickest one. You know that. But the important thing over here is that what is the use of capsule? What is the use of capsule is that all of you just listen to me. What is the important use of capsule is that, for example, let us say a bacteria who is having a capsule, it is entering into my body. Now, when the bacteria will enter into my body, it cannot die immediately, right? It has to survive. It has to survive. It has to divide. It has to affect the other organs, other tissues and all, right? A single bacteria, can it affect all the other organs? No. First of all, it will enter into your body. It will start dividing. Now, if it has to start dividing, don't you think your macrophages will see and attack it? But attack नहीं करना चाहिए. कैसे? These bacteria are having a structure called as capsule. This capsule prevents the phagocytes to come and eat them up. So can I call that these capsules are anti-phagocytic? They are acting as an anti-phagocytic. Yes or no? So what is the function of a capsule? Capsule कहीं पे भी देखो, any bacteria, if it is having a capsule, एक ही function होगा, anti-phagocytic. Okay. The function of a capsule is what? It is anti-phagocytic. Function of a capsule is anti-phagocytic. This is very, very, very important question. Clear all of you? So far, everyone is clear? Right. Next important thing. Next important thing is that you know that this is flagella. You know that this is flagella. Now, what I'm doing is that I'm just enlarging one flagella and I'll show it to you over here. Okay. Here they go. This is one flagella which I'm showing it to you here. Now, within the flagella, you have got some proteins. Actually, ये proteins नहीं है, ये actually antigens है. These are also called as antigens. Anyways, antigens are called proteins, right? So these proteins which are located within the flagella are called as H antigen. What is the name of this antigen here? This antigen is called as H antigen. This is very, 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 very important. Okay. What is the other name for this H antigen? It is also called as flagellar antigen. It is also called as flagellar antigen. So there are two important things. One is called as H antigen and other name for that is flagellar antigen. All of you are understanding, guys? Or am I only speaking to myself? Right? The new students, I know old students will definitely understand. The new students, I'm talking about the new students. Are you all understanding? Very good, very good. Chalo, let us go on to the next important thing. Next important thing over here is that, very good, very good. Right. Next important thing over here is that we have discussed what is the function of the flagella. You know what is the function of the flagella? It is responsible for locomotion. Not only that, flagella is also responsible for clearing the secretions and all, for movement and all. Okay. Chalo, leave it. But what is the function of pili? ये देखो यहाँ पे short hair-like structures, whatever are there, these structures I call them as pili. है ना? What is the function of pili? एक्चुअली दो तरीके के पाइलाई होता है. देर आर टू डिफरेंट टाइप्स ऑफ पाइलाई विच यू सी. ठीक है. व्हाट आर दीज टू डिफरेंट टाइप्स ऑफ पाइलाई आर दैट? सी, फॉर एग्जांपल, let me tell you one very important thing. Let us say this is a bacteria over here. ठीक है. Now this bacteria is entering into your body. This bacteria is having short process like this. These are called as pili. ठीक है. Now when this bacteria will enter into your body, first of all it has to settle somewhere, है ना. First of all it has to stick onto the host cell. ये host cell है. It has to stick onto the host cell. So if it has to stick onto the host cell, you need a protein here which will fix the bacteria to the host cell, ताकि ये bacteria does not move. So can I tell that pili, one function of pili is it is responsible for adhesion, right? So one function of pili is it is responsible for adhesion. ठीक है. दूसरा pili देखो. दूसरा pili क्या होगा? This is a bacteria which is having a pili. Okay. And let us, chalo, this is a bacteria having a pili. Now this is another bacteria having another pili. अभी ये दो बैक्टीरिया है ना, they wanted to conjugate. If you wanted to conjugate, what you need to do is that this bacteria which is having pili is going and fixing into another bacteria. So what has happened? A sex tube is formed. Through this sex tube, the genes from one bacteria will transfer to another bacteria. So what I'm trying to tell you is that I'm not trying to teach you how the sex of the bacteria happens here, but I wanted to teach you that one more function of pili is conjugation. Yes, one more function of pili is conjugation. So those pili which are responsible for adhesion, ये pili का नाम होता है common pili. These are called as common pili. ठीक है. और those pili which are responsible for conjugation, these are called as sex pili. So if I ask you how many different types of pili are there, very good, Krishna Bharadwaj, he is telling that transfer of genetic material between the cell. Very good. उसके लिए हमको कौन सा pili चाहिए? उसके लिए हमको sex pili चाहिए. Right? Now, these questions no one will ask you in the exam, but you know what they'll ask? They will ask you, ये sex pili कहाँ पे होता है? Sex pili are present for both gram positive bacteria. They are present for gram negative bacteria as well. But common pili are present only in gram negative bacteria. This is an important thing I want you to know, right? So regarding the pili, this is all you need to know. यहाँ तक, if you understood, raise your hand. Yes, 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 yes. Then I will go on to the next part. Understood? Just type कर लो. Yes. Very good. Very good, Ansari. Prashant, Vikram. Very good. चलो. Previously one question was asked. ठीक है. Previously एक question पूछा गया था. So what is this question is that they asked, they did not ask what is the antigen that is present in the flagella. You know antigen कौन सा है? H antigen. They did not ask that. They did not ask what is the function of the flagella. They simply asked gram positive bacteria में how many antigen rings are present. अभी ये antigen का rings क्या होता है? Just look here. If you look at the flagella, if you look at the flagella which is having H antigen, जैसे मैंने पहले ही बताया आपको, H antigen, right? If you look at the ultra structure, मैं PDF सेंड करूंगा. ओके, राज पटेल, आई विल डेफिनेटली सेंड द PDF. ठीक है, ग्रुप में PDF सेंड करें. So all of you just look at this. If you look at the structure of the flagella, this is how the structure of the flagella is. ये एक कंपोनेंट है फ्लैजेला का। अभी दूसरा कंपोनेंट ये है, ये दूसरा कंपोनेंट है, और इसके नीचे ये तीसरा कंपोनेंट है। सो हाउ मेनी कंपोनेंट्स ऑफ द फ्लैजेला डू वी हैव? वी हैव गॉट थ्री इंपॉर्टेंट कंपोनेंट्स ऑफ द फ्लैजेला। व्हाट आर दिस थ्री इंपॉर्टेंट कंपोनेंट्स ऑफ द फ्लैजेला? एनीवन फ्रॉम द प्रीवियस क्लासेस जो मैंने बताया था? Afsan Ansari, perfect. Your answer is right. Very good. So Afsan is telling that this part which is with the black is called as the filament of the flagella. This is called as the filament of the flagella. ठीक है? फिलामेंट के नीचे क्या होगा? ये छोटा सा पिंक कलर पार्ट जो है, दिस इज कॉल्ड एज अ हुक ऑफ द फ्लैजेला. व्हाट इज दिस? दिस इज कॉल्ड एज योर हुक ऑफ योर फ्लैजेला. हुक के नीचे क्या होगा? यहाँ पे चार रिंग्स देखने को मिल रहे हैं ना? देर आर फोर रिंग्स ओवर हियर. दीज फोर रिंग्स टुगेदर यू कॉल इट एज बेसल बॉडी. These four rings together you call it as basal body. सर ये रिंग्स का नाम क्या है? MPSL रिंग्स. इस रिंग्स का नाम क्या होगा? MPSL रिंग्स. ओके, फोर रिंग्स, MPSL. वेरी गुड अक्षय, वेरी गुड, वेरी गुड, वेरी गुड. MPSL रिंग्स. बट क्वेश्चन ये है, दिस वाज द क्वेश्चन विच वाज आस्क्ड. इन ग्राम नेगेटिव बैक्टीरिया हाउ मेनी रिंग्स आर प्रेजेंट? gram negative bacteria ka flagella, if you see within that flagella how many rings are present? The answer is all four rings are present. But if you look at gram positive bacteria, how many rings are present? Only two rings are present, और वो rings है M ring और S ring. ठीक है, M as well as S. Till here all of you are clear? Anis Momin, very good. Muskan Kumari, very good, very good, very good. All of you understood till here? Very good. Anand Prakash, very good. Nagesh, Paila Nagesh, you understood? Nagesh is here. Right. So the next important thing after this we will discuss is regarding the flagella. What they're going to give you is that flagell, एक bacteria का picture देंगे exam में. These are called image based questions. They'll give you one image of a bacteria and they will ask you, tell me what kind of flagella is this. ठीक है? What kind of flagella is this? First important thing. Now all of you just look at this bacteria and tell me यहाँ पे कितने flagella है guys? एक ही flagella है ना? So this is how the bacteria is, and there is one flagella onto one pole, है ना? So can I call it as monotrichous flagella क्योंकि एक ही flagella है? I am calling this flagella as monotrichous flagella. Monotrichous flagella. ठीक है? Sir, how do you remember this monotrichous flagella? There are two important examples. What are these two important examples? You need to know. First example goes is that Vibrio cholera. Vibrio cholera. All of you know Vibrio cholera, right? All of you know this. You have heard of this. Vibrio cholera is having a single flagella. और दूसरा worst organism है ये. इतना, you know, the infections are very dangerous. I will show you the pictures also. Infections are very dangerous, and they smell too badly. So this bacteria is called as Pseudomonas aeruginosa. The bacteria is Pseudomonas aeruginosa. So two important bacteria over here, guys. One is called as Vibrio cholera and the second one is called as Pseudomonas aeruginosa. These are the two important bacteria which are having monotrichous flagella. ये एग्जाम्पल्स कैसे याद रखना पड़ेगा. दो बार देखो, तीन बार देखो, चार बार देखो, सुबह सुबह उठ के देखो, कुछ भी कर लो, देख लो. सर एनी निमोनिक, एज ए निमोनिक इफ आई कीप ऑन गिविंग फॉर ऑल दी स्मॉल थिंग्स इट वुड बी अ हेल्प. ठीक है, जहां पे निमोनिक देना है वहां पे पक्का दूंगा. But now you just remember it as monotrichous flagella, Vibrio cholera and Pseudomonas aeruginosa. Till here you are clear? Very good. अभी ये देखो, what is the difference between the first bacteria and the second bacteria? First bacteria में तो एक ही flagella है, but यहाँ पे there are many flagella on to only one side of the pole. ठीक है? So such bacteria you call it as, such flagella you call it as Lopotrichous flagella. You call it as Lopotrichous. Monotrichous था पहले एक, अभी Lopotrichous flagella. ठीक है? एक एग्जाम्पल ऑफ लोपोट्रिकस फ्लैजेला इज योर स्पिरिल्ला. यू नो, स्पाइरल शेप्ड ऑर्गेनिजम्स जो है ना, पॉलीट्रिकस नहीं. अफसान, नॉट पॉलीट्रिकस. पॉलीट्रिकस मैं बता दूंगा व्हाट इज पॉलीट्रिकस. राइट, इट इज द परफेक्ट नेम दैट इज गिवन इज लोपोट्रिकस. Okay, so Nirav Agarwal, very good. Spirilla, you know, right? Spiral shaped organisms. I will show you the 3D animation. I mean, not animation, the real video of Spirilla later on. Okay, Muntazir, good evening. Right. Now, coming on to the third important thing. Third important flagella is, you see this bacteria over here. A bacteria के right side or left side, दोनों तरफ there is a flagella. So if two sides there is a flagella, can I call it as a half-half amphi? Yes or no? Very good, Tushar. This flagella is called as amphitricus flagella. Amphitricus flagella. So A for what? A for amphitricus. A also stands for a bacteria called as aquaspirillum. A also stands for a bacteria called as aquaspirillum. Now, A also stands for alkaligens. A also stands for alkaligens. Okay, alkaligens fecalis. Okay, sir, it is very difficult to remember these names. It is difficult. Okay, it was difficult for me also. So don't worry about this. Very good. Flagella located at the same spot. That is why it is called Lophotricus. Very good, Muskan. Very good. You're right. So far, how many flagellas we have discussed, guys? Only one flagella on one pole, monotrichus. From a single pole, many flagella are coming. That is called lophotrichus. On both the sides, 50% on the right, 50% on the left. This is called as amphitrichus. ये वाला amphitrichus है. Now I'm going to discuss one last one. A last flagella पता क्या है? ये देखो. Look at this picture. This is a bacteria. All over the surface of the bacteria, everywhere you have got this flagella, और इसका नाम होगा peritrichus. What is the name of this? This is called as peritrichus flagella. Peritrichus flagella. Okay. So where do you have this peritrichus flagella? I hope you know there is one gut bacteria which is very good for you. That is called as E. coli. Escherichia coli, you know that, है ना? Escherichia coli इतना अच्छा भी नहीं है ये बैक्टीरिया. I will also tell you about the worst outcomes of this bacteria later on. दूसरा एग्जाम्पल है, you will tell me the example, I will tell you the disease. ठीक है? एक डिसीज है जहां पे हमको टाइफॉइड आएगा. द नेम ऑफ द डिसीज इज टाइफॉइड. टेल मी व्हाट इज द नेम ऑफ द ऑर्गेनिज्म. जल्दी बताओ, पूरा नाम बताओ. निकिता, यू आर राइट. अफसान, यू आर राइट. तुषार टोल्ड मी दैट कंप्लीट नेम. योगमृता, वेरी गुड. साल्मोनेला टाइफ. वेरी गुड. Salmonella typhi. So Salmonella typhi causes the second important thing, and third important thing is Proteus. ऑर्गेनिज्म का नाम है प्रोटियस मिराबिलिस, बट ऐसे ही याद रखो कि प्रोटियस. ठीक है, जस्ट रिमेंबर इट एज प्रोटियस. वेरी गुड. सो गाइस, वी हैव डिस्कस्ड मोनोट्रिकस. ठीक है, उसके बाद सेकंड लोफोट्रिकस. ठीक है, उसके बाद एम्फीट्रिकस 50-50 ऑन बोथ द साइड्स. उसके बाद पेरिट्रिकस. दीज आर द 4 डिफरेंट टाइप्स ऑफ फ्लैजेला विच यू हैव. अगर यहां तक इफ यू अंडरस्टूड, आई विल टेल यू द नेक्स्ट फ्रॉम अभी से ना, राइट फ्रॉम नाउ ऑनवर्ड्स कॉन्सेप्चुअल पार्ट शुरू होगा. फ्रॉम नाउ आई विल स्टार्ट द कॉन्सेप्ट्स. तो यहां तक यू अंडरस्टूड, ऑल ऑफ यू. इफ यू अंडरस्टूड, देन आई विल मूव ऑन टू द नेक्स्ट इंपॉर्टेंट थिंग. जल्दी. All the 207 members in the hall, you understood. Everyone. New students. Old students are messaging. New students, batao. New students also, you're understanding, right? Amruta, MD Faiz, Pushpendra, Kavya, very good. Royal Ratna, you understood? Very good, very good. So today, what? Today is my first class. Okay, fine. Very good, Tushar. Chalo. If you're understanding, then that is very good for me also. Chalo. Next important thing is that, okay, you are telling flagella, ठीक है, flagella, एक flagella है, दो flagella है, one side flagella, both sides, everywhere flagella. चलो ठीक है. how will you identify the flagella? That is a very important question. How will you identify a flagella? So how do you identify a flagella? How will you see a flagella? So all of you guys put your pens down. Don't write anything. Don't write anything. Flagella, I can look by two ways. Okay? There are two methods how I look at a flagella. Very simple. एक method, बैक्टीरिया उठाओ ऐसे स्लाइड के ऊपर रखो, माइक्रोस्कोप, look from the microscope, right? You will look at the bacteria and you will look at the flagella. इसको क्या बोलते हैं? इसको बोलते हैं डायरेक्ट मेथड, है ना? You are looking directly from your naked eye by using microscope. That method is called as a direct method. और दूसरा मेथड है वो क्या होगा? इनडायरेक्ट मेथड. सो दो मेथड्स है टू लुक एट अ बैक्टीरिया. वन इज कॉल्ड एज अ डायरेक्ट मेथड और दूसरा क्या होगा? इनडायरेक्ट मेथड. अनदर मेथड वुड बी इनडायरेक्ट मेथड. ओके ना? सो डायरेक्ट मेथड and indirect method. How do you look at direct method? In direct method, what do you use? You use what? You use electron microscope. You use electron microscope in direct method. Now, in indirect method, what do you see? You use multiple things in indirect method. I'm going to tell you one by one. Just note it down. First important thing. First important thing क्या है कि I will take, ये देखो, all of you now put your pens down. Just listen to whatever I'm telling you. ठीक है? What I will do is that I will take a glass slide. Glass slide जानते हो ना? लैब में जो मिलता है ना? When you go to the hospital, doctors do a blood test on one slide, है ना? उस टाइप का ग्लास स्लाइड. बट ये ग्लास स्लाइड में ना बीच में इन द सेंटर ऑफ दिस ग्लास स्लाइड देयर इज अ स्मॉल डिप्रेशन. ठीक है? फॉर एग्जांपल, मान लो ये ग्लास स्लाइड है. ओके, दिस वाज योर ग्लास स्लाइड. ठीक है? ये ग्लास स्लाइड के बीच में इन द सेंटर यू सी अ स्मॉल डिप्रेशन हियर. ठीक है? नाउ व्हाट आई एम डूइंग इज दैट आई विल टेक ए कवर स्लिप. ठीक है? एक मान लो एक अल्यूमिनियम फॉइल. अल्यूमिनियम फॉइल, छोटा सा फॉइल ऐसे कट कर दिया मैंने. आई हैव टेकन अ स्मॉल अल्यूमिनियम फॉइल. आफ्टर टेकिंग दिस अल्यूमिनियम फॉइल, व्हाट आई विल डू इज दैट आई विल ऐड फ्यू ड्रॉप्स ऑफ बैक्टीरिया. ठीक है? टेस्ट ट्यूब में जो बैक्टीरिया है, आई विल टेक दैट बैक्टीरिया एंड आई विल ऐड फ्यू ड्रॉप्स ऑफ बैक्टीरिया ऑन टू दिस अल्यूमिनियम फॉइल. अब मैं क्या करूंगा? आई विल पुट दिस अल्यूमिनियम फॉइल अपसाइड डाउनवर्ड्स लाइक दिस. ठीक है? सो इमीडिएटली आई विल टर्न इट. व्हेन आई टर्न इट यू माइट थिंक दैट ये ड्रॉप जो है, ड्रॉप के अंदर बैक्टीरिया जो भी है, ऑल दिस ड्रॉप माइट फॉल डाउन, बट दिस ड्रॉप विल नॉट फॉल डाउन क्योंकि सरफेस टेंशन बहुत है यहाँ पे. राइट? सो दिस ड्रॉप विच यू आर एडिंग हियर इज कॉल्ड ऑयल ड्रॉप. ऑयल का सरफेस टेंशन ज्यादा होगा, है कि नहीं? सो विद इन दैट ऑयल ड्रॉप देयर इज बैक्टीरिया. आई एम टर्निंग इट. व्हेन आई एम टर्निंग इट क्या होगा? ये ऑयल ड्रॉप जो है ऐसे हैंग होगा, है कि नहीं? स्प्रिंग जैसे हैंग होगा, है कि नहीं? नाउ जस्ट लुक एट द पिक्चर विच आई एम पुटिंग ओवर हियर. दिस इज दैट कवर फॉइल. ठीक है? नाउ व्हाट आई एम डूइंग इज दैट आई एम पुटिंग दिस कवर फॉइल ऑन टू दिस डिप्रेशन. और ऑयल ड्रॉप कहां पे है? ये देखो, ये है ऑयल ड्रॉप. दिस वॉज द ऑयल ड्रॉप विच इज हैंगिंग. और इसके अंदर क्या है? इसके अंदर ये छोटे से रेड कलर थिंग्स विच आई एम ड्रॉइंग, दीज आर द बैक्टीरिया विच आर प्रेजेंट. ठीक है सर? ऐसे तो दिस ऑयल, दिस फॉइल वॉट एवर इज देयर, ये तो नीचे गिरेगा. इसीलिए व्हाट आई विल डू, ऑन बोथ द साइड आई विल पुट ए स्टैंड. ये स्टैंड का नाम है पेट्रोलियम जेली. पेट्रोलियम जेली. ठीक है? नाउ व्हाट आई विल डू इज दैट, नाउ व्हाट आई विल डू इज दैट विद इन द डिप्रेशन दैट ऑयल ड्रॉप विल बी हैंगिंग. है ना? मैं एक साइड लाइट पास करूंगा और दूसरा साइड से आई विल लुक एट द बैक्टीरिया, हाउ द बैक्टीरिया आर मूविंग. है ना? आर द बैक्टीरिया आर मूविंग इन दिस वे स्पाइरली, और द बैक्टीरिया आर मूविंग अप एंड डाउन, और द बैक्टीरिया आर falling like a leaf. है ना? When the leaves fall down from the tree, you know how they fall down? Is it falling like a tree? Right? Is the bacteria shaking? So all the motility of the bacteria, I can look in that oil drop. I hope you all are imagining whatever I'm telling you right now. Right? So this experiment, whatever I have shown you over here, is called as hanging drop experiment. क्यों हैंगिंग? क्योंकि ये ऑयल ड्रॉप जो है ये हैंग हो रहे हैं ना. दैट इज यू कॉल इट एज हैंगिंग ड्रॉप एक्सपेरिमेंट. हैंगिंग ड्रॉप एक्सपेरिमेंट. ठीक है? सो वन इज विद द हेल्प ऑफ दिस हैंगिंग ड्रॉप एक्सपेरिमेंट आई कैन लुक हाउ द मोटिलिटी ऑफ द बैक्टीरिया इज टेकिंग प्लेस. सेकंड एग्जाम्पल, सेकंड एग्जाम्पल ये है कि अनदर एग्जाम्पल इज वेट माउंट प्रिपरेशन, वेट माउंट preparation. Second important thing is wet mount preparation. Third important thing is dark field microscopy. Third important thing is dark field microscopy. And fourth important thing is phase contrast microscopy. All of you know the names of these microscopes, right? एक होगा सिंपल माइक्रोस्कोप, एक होगा कॉम्पाउंड और सिंपल डार्क फील्ड, एक होगा फेज कॉन्ट्रास्ट, एक होगा इलेक्ट्रॉन, है ना? फेज कॉन्ट्रास्ट माइक्रोस्कोपी. So why did you write these four things? With the help of these four things, I can look at what? I can look at the motility of the bacteria. I can look at what? Motility of the bacteria. Tomorrow in your classes, if your microbiology teacher there in Bishkek, if she's asking you how you can look at the motility of the bacteria, four important things you need to tell. Hanging drop experiment, wet mount preparation, dark field microscopy, phase contrast microscopy. Even if you are telling the right answer, वहां पे कुछ और ही आंसर होगा. So यहाँ पे जितना whatever you have to learn, just learn it, ignore them. ठीक है? So hanging drop, wet mount, dark field, and phase contrast. Sir, how do you remember this? How do you remember this? Is that you love to explore in the night? You love to hang out with your friends in the night or in the daytime? बताओ. Most of you love to hang out with your friends in the night. Okay. So we hang out in the night. Night को क्या बोलते हैं? Can I call it as dark phase? है ना? Can I call it as dark phase? So you love to hang out with your friends in the dark phase. ठीक है? V क्या होगा यहाँ पे? V क्या होगा? V is nothing but wet mount preparation. Hang क्या होगा? Hang मतलब hanging drop experiment. Third important thing is dark. What is dark? Dark field microscopy, and phase is phase contrast microscopy. So these are the four ways how you remember the motility testing of a bacteria. All of you understood? Before I move on to the next one, I want only the new students to answer. है कि नहीं? जल्दी बताओ. यहाँ से 2017, I think 2017 there was one FMG question that was asked continuously for 3 exams, मतलब 1.5 year continuously the question was asked from this. All of you are understanding? Very good. चलो. Now, sir, you are telling me that bacteria का motility हम देखेंगे, right? What kinds of motility do you see? What are the different kinds of motility? You told me one motility, अगर बैक्टीरिया ऐसे मूव हो रहा है, दिस इज कॉल्ड एज स्पाइरल मोटिलिटी. ठीक है? ये छोड़ के बाकी क्या क्या मोटिलिटी है? व्हाट आर द डिफरेंट मोटिलिटीज? You just have to remember this, right? From this also a question was asked previously, very 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 important one. So different types of motility, एक होगा tumbling motility, एक होगा darting motility, एक होगा swarming motility, एक होगा falling leaf motility. Falling leaf motility मतलब I will show you the videos later on. ठीक है? Falling leaf मतलब जैसे from the tree as the leaf falls down, will the leaf fall down like this? No, the leaf will turn depending upon the air turbulence and it will fall down like this, right? Bacteria भी ऐसे ही गिरेगा नीचे. Next, corkscrew motility and lashing motility. Lashing मतलब ऐसे, lashing. Okay, so these are the different motilities. Now, what is tumbling motility? ठीक है? Tumbling motility is given by which bacteria? ये देखो, this is T. T I can write it as L. है कि नहीं? L stands for what? Listeria. And the organism called as Listeria monocytogenes. You need not to remember the total name. ये याद रखो बस. Listeria is responsible for tumbling motility. Okay. Now, second important motility, darting motility. ठीक है? Darting मतलब क्या है? Darting मतलब you are darling. Okay, so whenever your darling is beside you, you will have positive vibes or you will have negative vibes, guys. बताओ. जो experienced बंदा है यहाँ पे वो बताएगा. When your darling is behind you, you will have positive vibes or you will have negative vibes. प्रशांत विक्रम गिरी, पॉजिटिव वाइब्स. तो अभी अभी रिलेशनशिप में अंदर आया होगा तभी लग रहा है तुमको पॉजिटिव वाइब्स. अथर्व इज आंसरिंग द राइट आंसर, दैट इज नेगेटिव वाइब्स, है ना? मॉय मॉय नहीं रे भाई. पॉजिटिव. पॉजिटिव वाइब्स की नेगेटिव वाइब्स. नेगेटिव वाइब्स. ठीक है? नेगेटिव वाइब्स. नेगेटिव. नेगेटिव में वीई है. यस और नो? नेगेटिव में वी ही है। वी स्टैंड्स फॉर विब्रियो कोलेरा। वी स्टैंड्स फॉर विब्रियो कोलेरा। ठीक है, वी स्टैंड्स फॉर विब्रियो कोलेरा। सो डार्टिंग मोटिलिटी इज डन बाय विच बैक्टीरिया ओवर हियर? विब्रियो कोलेरा। ठीक है। स्वॉर्मिंग मतलब क्या होता है? स्वॉर्मिंग मतलब स्विमिंग। इफ यू वांटेड टू लर्न स्विमिंग, यू विल गो टू ए पर्सन हु जॉइंड टुडे, और यू विल गो टू ए प्रो स्विमर। प्रो स्विमर के पास जाएगा, है ना, सो दैट ही विल टीच यू। सो pro, you will go to a pro swimmer. Yes or no? If you wanted to learn swimming, you will go to a pro swimmer. Pro क्या होगा यहाँ पे? Pro in the sense Proteus. Pro in the sense Proteus. I think we discussed Proteus somewhere. कहाँ पे डिस्कस किया था Proteus? ये देखो Proteus. Proteus is having what kind of flagella? Peritrichous flagella. Okay, it is having peritrichous flagella. So Proteus is swarming motility. ये याद रखो, Proteus का कौन सा मोटिलिटी है? Swarming motility. What flagella? Peritrichous flagella. Next, falling leaf motility. Falling, for example, if a person is falling from 10th floor, वो रहेगा कि गया? He is गया, right? He is dead. So गया मतलब क्या? गयाटिया, right? So just remember it as gaya in the sense gyatia. Remember? You'll remember it. Gyatia. So gyatia is responsible for falling leaf motility. Clear? Now, next important thing is, if you guys wanted to go on a trip, okay, long trip with your friends, all of you will book an auto and go to a trip, or will you book a car? If you have a car, that is fine. So you guys will take a car for your trip. कार मतलब क्या? कॉर्क. व्हाट इज कार? कार इज नथिंग बट कॉर्क. ट्रिप मतलब क्या? ट्रेपोनिमा. ट्रिप इज नथिंग बट ट्रेपोनिमा. ऑल ऑफ यू नो ट्रेपोनिमा पैलिडम, है ना, सिफिलिस. अहमद विल टेक मशूद का. वेरी गुड अहमद. मैं इंडिया में मशूद का नहीं है ना. सो corkscrew motility is done by which bacteria here? Treponema pallidum. Treponema. Okay. Now lash मतलब क्या? Lashing. So whom do you lash? Whom do you lash? बताओ, whom do you lash? ज्यादा आउट ऑफ द बॉक्स नहीं सोचना. whom do you lash मतलब, in the villages you lash buffaloes, है ना? You lash basically buffaloes. You lash a buffalo, right? Now what I'm telling you, buffalo is also called as borrelia. Okay, so remember buffalo as borrelia. So lashing motility कहाँ पे देखोगे? In borrelia you see lashing motility. All of you understood whatever I've told you, है ना? ये इसमें कोई कॉन्सेप्ट नहीं है, इसको ऐसे ही याद रखना पड़ेगा. That is why I've taught you like this only. So tumbling motility is in Listeria, darting motility Vibrio cholera, swarming is Proteus, falling leaf is Giardia, corkscrew is Treponema, and lashing is your Borrelia. Everyone understood till here? यहाँ तक समझ में आ गया सबको? Everyone, न्यू स्टूडेंट्स, न्यू स्टूडेंट्स, ठीक है. वेरी गुड, वेरी गुड, वेरी गुड, वेरी गुड. मुस्कान, मेहमान, MD5, वेरी गुड. ओके, राइट. Next important thing over here is that after discussing this, the next important part we have to discuss is spore. पता, never ever underestimate a spore. Every year microbiology out of 17 questions, एक question तो पक्का spore से आएगा. If you understood whatever things I'm teaching you right now in spore, 1 mark is right now in your pocket. Okay. So what things can you discuss about a spore? What are the things that you can discuss about a spore? एक चीज याद रखो कि स्पोर्स जो है ना, दे आर प्रेजेंट इन ओनली ग्राम पॉजिटिव बैक्टीरिया. सर ये ग्राम पॉजिटिव ग्राम नेगेटिव क्या है, मैं बाद में बताऊंगा. ओके. नाउ you have to remember, spores even live for 1,000 years. Spores even live for 2,000, 3,000 years also. You remember that? 2,000, 3,000 years के बाद you take that spore, add water to it, provide some favorable climate, environment and all, फिर से वो spore active हो जाएगा, है ना? Spores are that very very resistant. इसका मतलब क्या है? Can I tell that spore is resistant to heat? है ना? Spore is resistant to dehydration also. and spore is resistant to chemical also. So chemically, heat wise, temperature wise, and dehydration wise also, spore is very, very, very stable. Yes, Afshan Ansari, very good. Spore tolerates to adverse conditions. Now what I'm telling you here is that I will end up this class very fastly, guys. Don't worry, okay? Don't worry about that. First class I will end up it here, and उसके बाद I know मेस का टाइमिंग हो रहा है ना? एक दिन एक दिन थोड़ा लेट खाने से कुछ नहीं होने वाला है. कल से ये आदत बनाओ कि थोड़ा लेट खा लो, ठीक है? So coming to the spore gram positive bacteria only. Now, one very important thing you need to know is that there are only two species which release spores. ये याद रखो. Only two species which release spores. One is called as Bacillus और दूसरा होगा Clostridium. All of you know Clostridium tetani, Perfringens, Botulinum है ना? So Bacillus and Clostridium सिर्फ ये दोनों ही species है which release spores. Okay. So what are the two species that release spores? One is called as Bacillus. Another one is called as Clostridia. These are the only two species which release spores. ठीक है? These are the only two species which release the spores. Now, next important thing is that, sir, you are telling spores can live for about 1,000 years, 2,000 years. What is making कैसे? How can a spore live for 2,000 years? मतलब उसके अंदर कुछ होना चाहिए ना? There should be something within the spore which is making the spore to live for 1,000 to 2,000 years. So what is that thing in the spore? First important thing. If you look at a spore, स्पोर का सेंटर पार्ट है ये. दिस इज कॉल्ड एज अ कोर. सेंटर पार्ट क्या होगा? दिस इज कॉल्ड एज अ कोर. नाउ सराउंडिंग द कोर यू सी अ रेड कलर लेयर, है ना? सराउंडिंग द कोर दिस पर्टिकुलर रेड कलर लेयर इज कॉल्ड एज स्पोर कोट. इसका नाम क्या है? इसका नाम है स्पोर कोट. मुंताजिर शाफी, यू आंसर्ड इट वेरी करेक्टली, वेरी गुड. दिस इज कॉल्ड एज स्पोर कोट. स्पोर कोट के surrounding the spore coat also you have got a double layered structure like this. ठीक है? This is called spore wall. इसको क्या बोलते हैं? Spore wall. Spore wall के surrounding सिर्फ एक ही layer है. This is called as exosporium. What is this called? Exosporium. So let us write down the things. इसका नाम है exosporium. इसका नाम क्या है? This is called exosporium. और ये पिंक कलर में जो मैंने ड्रॉ किया, दिस इज कॉल्ड एज स्पोर वॉल. स्पोर वॉल. और ये रेड कलर में जो है, दिस इज कॉल्ड एज स्पोर कोट. और एक चीज आई हैव टू राइट इट हियर, दैट स्पोर कोट और स्पोर वॉल के बीच में यू सी दिस येलो कलर लेयर विच आई एम ड्राइंग, राइट? दिस इज कॉल्ड एज कॉर्टेक्स ऑफ द स्पोर. व्हाट इज दिस कॉल्ड? इसका नाम है कॉर्टेक्स. इसका नाम है कॉर्टेक्स. सो टिल हियर यू अंडरस्टूड, राइट? Now, from this spore, I told you every year one question will be asked. Two questions are asked previously. एक question है, spore coat is made up of what? What is spore coat is made up of? Someone already answered. Tushar answered it rightly. Spore coat is made up of keratin. Okay, spore coat is made up of keratin. Very, very, very important thing. Spore coat is made up of what? Keratin. Out of all layers, which is the thickest layer? ये question भी पूछा गया था. What was the thickest layer? Your cortex is the thickest layer. Your cortex is your thickest layer. Third important thing is that, sir, you told me that spore will live for 1,000 years. How? How it is living for 1,000 years is that ये core of the spore है ना, ये core के अंदर एक acid है. There is one chemical within the center of this core. और इस chemical का नाम anyone will tell? You have already answered this. Anyone? Yes? Anyone willing to answer? Very good. Very, 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 very good. Kishan, diplomacy nahi. Diplomacy, politics nahi. Muntazir, group one. Very good. Very good, Bhavika. So this acid, this is called as dipicolinic acid. Dipicolinic acid. So what is the acid? Dipicolinic acid. Dipicolinic acid के वजह, it is because of dipicolinic acid, your spore is able to live for many, many, many years. This thing I want you to know. ठीक है? I will also show you and tell you how dangerous these spores are. ठीक है? Just look at the screens. All of you just look at the screen over here. Everyone look at the screen. What did I tell you? I told you only two species are responsible. They release the spores. Bacillus और Clostridia. ये देखो. Bacillus में Bacillus anthracis है. Bacillus cereus. These are the two organisms. In Clostridium, you have got four organisms. One is called botulinum. You know botulinum. You know tetany also. Only two new things you have to know. एक है Clostridium difficile and Clostridium perfringens. Clear? Can anyone tell spores of Bacillus anthracis will cause what? What will the spores of Bacillus anthracis cause? कौन सा disease cause करेगा? Very famous है. There was an outbreak also. Very good, Fatima. Muntazir, Tushar, perfect. It would cause what? It would cause anthrax. It would cause anthrax. Bacillus cereus ka spores will lead to what? What does it cause, Mahindra? Very good. Very good. Excellent. They are going to cause food poisoning. They are going to cause food poisoning. Next, botulinum. You heard of this 1,000 times, I think. Botulinum will cause botulism. वो भी एक type का food poisoning है, right? That would cause botulism. Next, Clostridium difficile will cause what? This is a very important thing which few members know. Very good. Very good. Very good. So very good. Clostridium difficile will cause PMC. PMC मतलब pseudomembranous colitis. Pseudomembranous colitis. It would cause what? Pseudomembranous colitis. Next, Clostridium perfringens. Perfringens पता क्या करेगा? It will perforate deep inside your tissues. Bacteria पता क्या होगा? Bacteria will go deep inside the tissue. That is why the name is perfringens. It will perforate deep inside the tissues. Bacteria अंदर तक जाएगा and it will start releasing the gas. When it starts release the gas, क्या होगा? This area will completely be swollen. और ये swollen area के अंदर क्या होगा? पूरा बैक्टीरिया का गैस होगा ना? What gas? हाइड्रोजन सल्फाइड होगा. All of you know. That is why you call this Clostridium perfringens causing agent is gas gangrene. It would cause what? Gas gangrene. Okay. Finally, Clostridium tetanus will cause tetany. Now just look at the pictures. ये देखो. All of you look at these pictures. You see what is this particular structure over here? ये पता है ये क्या है? What is this black color thing, you know? Is this pus or is this gas? Gangrene है, very good. Gangrene, you're right. इसका नाम क्या है? Gas gangrene. The gas gangrene smell is too worst. Too worst because बैक्टीरिया का हाइड्रोजन सल्फाइड जो गैस रिलीज होता है वो गैस दैट यू हैव टू स्मेल एंड दिस इज द गैस गैंग्रीन. और यहाँ पे ये देखो व्हाट इज दिस गैंग्रीन हियर? ये पूरा ड्राई हो गया ना, है ना? दिस इज कॉल्ड ड्राई गैंग्रीन. This is called dry gangrene and this is called gas gangrene. Clear all of you? Gas gangrene, you know what is the bacteria that is causing? Clostridium perfringens. But dry gangrene, any idea, anyone? कौन सा disease में, in which disease will you able to see dry gangrene? बताओ. Which disease will you see dry gangrene? In which disease will you see dry gangrene? Anyone, any idea? Very good, Nidhi Jain. Very good, very good. Excellent, Fatima. Very good. Ajit Kumar. Dry gangrene you see in diabetes. If a patient who is having diabetes mellitus type 2, okay, and if is not at all controlled, उसका diabetes controlled status में नहीं है, it is not at all controlled, right? So in that case, what will happen is that the glucose starts depositing within the blood vessels. तो क्या होगा? Blood vessel block हो गया. अगर blood vessel block हुआ, do you think blood will be supplied to your legs? No. If blood will not be supplied to your legs, patient will have dry gangrene. The entire area will become dry and it will fall off. Even for the hands also, patient can have dry gangrene. Okay, right. Now look at this picture. You know, what is this picture over here? ये बताओ ये क्या है? What is this? Dry gangrene or wet gangrene? Yes. He is asking a very good doubt. Rahul Kumar, reason क्या है gangrene का? That entire mechanism I will explain you when I will be teaching this Clostridium. ठीक है? तब explain करूंगा अच्छे से. See, this is your wet gangrene. राइट, वेट गैंग्रीन, क्योंकि ये देखो पूरा वेट है ये एरिया, कैन यू सी, कंप्लीट वेट. सो लेटर ऑन व्हाट विल हैपन, स्लोली यहाँ पे गैस डिपोजिशन हो रहा है एंड इट विल टर्न टू गैस गैंग्रीन. क्लियर ऑल ऑफ यू? राइट. So this was a patient who was having gangrene. ठीक है. So हमने क्या किया यहाँ पे, we have removed all that gangrenous part. यहाँ पे देखो, within the dustbin you can see all this black color thing, है ना. This is the removed of gangrenous part. हम इसको कट करना पड़ा, पूरा कट करने के बाद this is how his leg was. Okay, so this is a picture of your dry gangrene over here. Clear? So this was a picture of your dry gangrene. Got it, guys? All of you understood? Everyone perfectly understood whatever I've taught. So guys, I won't trouble you much today. Pehla class hai na. So let us stop it here. There are a lot of memory things which we have to discuss over here. In next class I will teach you the complete mechanisms. Bacteria कौन सा toxins release करेगा, okay, कौन सा toxins release करेगा, and what will happen with these toxins, what is the mechanism, endotoxic shock क्या होता है. उसके बाद what we will do is that we will also discuss the multiple choice questions which was asked previously. Okay, क्या हो गया कृष्णा सर, ओपी, ओपी मतलब टॉप. टॉप क्या है टॉप? टॉप क्या होगा, बिकॉज टुडे टाइमिंग वाज 7 o'clock इन द बिशकेक टाइम. ओवर पावर, ओवर पावर्ड, व्हाट इज ओवर पावर्ड? ओवर पावर्ड मतलब कुछ प्रॉब्लम हुआ. फाइन. अच्छा अच्छा, इट मीन्स गुड. ठीक है, थैंक यू. फाइन फाइन, आई अंडरस्टैंड. Offline, I'll try to come, guys. Right now, the schedules are very busy, so we are right now concentrating only on the online sessions. But offline, I will try to discuss. And ये देखो, यहाँ पे मैंने जो questions दिखाया था, these questions were previously asked. ये questions भी हम discuss करेंगे. And obviously, as usually, we will discuss everything conceptually. ठीक है? So tomorrow class, कल भी class same time शुरू होगा. Same time 6:30. Okay. If you want any change in the timings, you can talk to the management. क्योंकि मैं रेस्पॉन्सिबल नहीं होगा ना, ये मिस टाइमिंग का मुझे थोड़ी लेना देना है. सो ठीक है. सो ऑल ऑफ यू गाइस, आई विल शेयर दिस पीडीएफ राइट नाउ इन द ग्रुप, टेलीग्राम ग्रुप. एंड आई विल आल्सो शेयर यू माय टेलीग्राम ग्रुप आल्सो, सो देयर आल्सो यू कैन फाइंड आउट द नोट्स. Yes, we will also do that here in India only, anatomy also. Okay, so all of you guys, you understood all the things, you understood all the things in the class, everything whatever I've taught you so far. In the group we will tell you the telegram. What is the name of the telegram? You understood? Very good. So, guys, thank you so much. Thank you so much for your patience. So, goodbye, take care, and love you all.